Para Science Hall. Your victory is our responsibility. Proper guidance of medical engineering undergraduates.
Okay, today we are going to study about nucleus. What's the main function of nucleus? Control the cell activity. Okay. When you consider about human body, which part is equal to nucleus in cell? Normally, when you consider about human body, which part is controlling human body? Brain. So likewise, when you take the cell, nucleus is the very important part. Okay. Normally, what's the shape of the nucleus? Spherical shape. Nucleus is made up of two membranes. Okay, two membranes. Outer one is outer membrane and inner. Okay, outer membrane and inner membrane. Outer membrane and inner membrane. Between these two membranes, there is a small gap. Value of that gap is 20 to 40 nanometers. Okay, 20 to 40 nanometer. Normal diameter of the nucleus is 5 micrometer. Okay, so diameter of the nucleus 5 micrometer. Nucleus is composed of two membranes. Outer one is outer membrane and inner membrane. Between outer and inner membrane, 20 to 40 nanometer gap is present. Then inside the nucleus, there are two main parts. This one is known as nucleolus. Okay, nucleolus, and this one DNA-like structure is known as chromatin. Okay, what's heading? Nucleus. Main function of nucleus: controls the activity of cell. Nucleus is composed of two layers: outer membrane, inner membrane. Between outer membrane and inner membrane, there's a small gap, 20 to 40 nanometer. After that, there are two main parts: nucleolus and chromatin. Nucleolus is mainly made up of RNA and protein. Next one, chromatin. Chromatin is made up of DNA. Okay. What is the meaning of chromatin? A group of DNA molecules. Okay. Group of DNA molecules are known as chromatin. Okay. Other than this, there are small pores present in the membrane of the nucleus. So these pores are known as nucleus pores. Okay. The function of nucleus pores is, what's the main function of plasma membrane? Controls the entry and exit of the substances. So likewise, so here also controls the entry and the exit of the substances. Okay. So how many points you have to tell? According to the structure of nucleus, composed of two membranes. First one, inner membrane, outer membrane, gap 20 to 40 nanometers. After that, what's the diameter of the nucleus? 5 micrometers. Okay. Next one. What? Yeah, two parts. First one, nucleolus and chromatin. Chromatin means a group of DNA. Nucleolus is made up of RNA and proteins. Okay. Main function of nucleolus controls the cell activity. Other than that, protein synthesis. Okay, protein synthesis. Okay. Now tell 5 micrometers, how many nanometers? 5 micrometers, how many nanometers? Sometimes you will get questions like this. Okay, micro means 10 to the power minus 6. Micro. Nano? Very good. Minus 9. So different? 3. So 5 micrometers is equals to 5000 nanometers. So 5000 nanometer diameter. What's the gap? 20 to 40 nanometer. Clear? Okay. Now read your resource book. What's the name of this? Liquid-like area. Fluid. Nuclear. Nuclear matrix. Okay, nuclear matrix. Okay, read below. Who can read? Can you?
Has been a transmitter of 5 micrometers. Nucleus nuclear. Nuclear consists of two membranes, inner and outer membrane.
Yeah. Wait. So inner membrane, outer membrane. Altogether known as nuclear envelope.
Separated by a space of 20 to 40 millimeters. Nuclear envelope is perforated by nuclear bonds which have 4 surface rays reducing energy having of outer. It has a nuclear laminate made of protein filaments which lie in the exterior side of the nuclear envelope.
Yeah. This interior part of the nucleus is made up of protein filaments. What is meant by filament? Filament means fibers-like structure. Okay, fibers-like structure. How to convert chromatin into chromosomes? Tightly. Tell. Tightly coil. First point. Tightly coil. Forms thread. Forms thread. In human species, how many chromosomes are there? 23 pairs. How many chromosomes I have? 23 pairs. How many sexual chromosomes? 1 pair. Or 2 pairs. No, sexual chromosomes. How many sexual chromosomes are there? 1 pair. In male? Male? Female? No, no. Maybe you didn't study it. In male XY, female XX. Okay? Okay? Let us study, okay? So, this is the sexual chromosomes. Okay, in human species, 46, 23 pairs. In other species, same or different? Different, okay? Only that question? Now tell, which human cell contains 23 chromosomes? Which human cell contains 23 chromosomes? Normally, all the human cells are composed of 23 pairs of chromosomes. But there's a cell, it contains only 23 chromosomes. Don't know. No idea. Okay. Those cells are known as gametes. Gametes. Don't know. Okay, maybe you may don't know. Sperm and egg. Okay. These are little advanced things. Okay. You don't know about these things. 23. This is advanced. O level. Not O. Grade 10. Okay, grade 10 syllabus. Okay. First one. Nucleus is composed of envelope, nuclear envelope. Nuclear envelope contains two membranes, outer membrane, inner membrane. Between these two membranes, there is a membrane gap. What's the value? 20 to 40 nanometer. What's the diameter of the nucleus? 5000 nanometers, so 5 micrometers. Next one. Nuclear envelope composed of spores. What is the function of spore? Allows the exit and entry and the exit of the substances. Next one. Two main. Nucleolus, chromatin. Nucleolus made up of RNA and protein. Protein or protein filament. Next one. Okay. How chromatin turns into chromosomes? It gets tightly coiled and forms threads-like structure. Normally, when you consider the human species, how many chromosomes are there? 23 pairs of chromosomes. Very good. Next one. Nucleus is filled with nuclear matrix. Okay. Only this question? Okay. Now function. Okay, ribosome. Ribosome is composed of two subunits, larger subunit and smaller subunit. Larger subunit and smaller subunit. Okay, larger one and smaller one. Okay, larger subunit and smaller subunit. So which one is larger? Upper one, smaller one. Okay, very simple part. Okay, these are the two subunits of ribosomes. Other than that, there are two types of ribosomes. Under eukaryotes we studied 70S and 80S ribosomes. 70S ribosomes and 80S ribosomes. 70S ribosomes present in where? Prokaryotes and eukaryotes. 80S only present in eukaryotes. Okay, these are the two types of ribosomes according to the structure. According to the structure, ribosomes divide into two. According to the location, again ribosomes divide into two. Free ribosomes, membrane bound ribosomes. So there are two types of two subunits are there, smaller subunit, larger subunit. According to the structure, ribosomes divide into two, 70S ribosomes, 80S ribosomes. 70S ribosomes present in both prokaryotic and eukaryotic cells. 80S ribosomes present only in eukaryotic cells. According to the location, ribosomes divided into two, free ribosomes and membrane-bound ribosomes. Mostly, you can see this membrane-bound ribosome in RER, rough endoplasmic reticulum. Okay? Free ribosome mostly present in cytoplasm. What is the physical state of cytoplasm? Solid or liquid or semi-solid? Very good. Semi-solid. Okay? So these are the main points about ribosomes. What is the main function of ribosome? Protein synthesis. Ribosomes are formed from nucleolus. Okay, nucleolus. Because ribosomal subunits are created by whom? Now only said rRNA and ribosomal subunit. Under which part? Functions of nucleolus. Okay, functions of nucleolus. What is the function of nucleolus? Synthesize RNA and ribosomal subunit for protein synthesis. Okay, for protein synthesis. So, ribosomal subunits are made by nucleolus. Clear, Mahdi? Okay. Functions of ribosomes. They are composed of RNA and protein. Same as nucleolus. Where you can see 80s ribosomes? 70s ribosomes? Cytoplasm in prokaryote and matrix of mitochondria. Stroma of chloroplast. Matrix means fluid-like structure. Stroma means what is the function of chloroplast? Photosynthesis, production of chlorophyll. So it will roughly look like this. So this area is known as stroma. But same thing in mitochondria. This is mitochondria, rough structure of mitochondria. So this area is known as matrix. But here we are not using the word matrix instead of stroma. Okay. Now, but according to your resource book, there's a small difference. 80S ribosomes divide into two. 80S ribosomes, according to the location, divide into two. Free and membrane bound. Free present in cytoplasm. Membrane bound present in rough endoplasmic reticulum. There are two types of endoplasmic reticulum, rough and smooth. Why rough endoplasmic is known as rough endoplasmic reticulum? Why? Because the surface is rough. What's the reason? Because the attachment of ribosome. Ribosome, main function of ribosome, protein synthesis. Ribosome is composed of two subunits, larger subunit, smaller subunit. There are two types, 70S, 80S. 70S ribosomes present in cytoplasm in prokaryotes, matrix in mitochondria, stroma in chloroplast. 80S ribosomes present according to the location. 80S ribosomes divided into two, free ribosomes and membrane bound ribosomes. Free ribosomes present in cytoplasm of eukaryote or prokaryotes. Eukaryotes. 80S. No, 80S, okay? Next one, membrane bound ribosomes are attached in rough endoplasmic reticulum. Subunits are composed of rRNA and protein. Function of ribosome. Protein synthesis. Clear? Okay, I am. Why silent today? Why silent today? Any problem? No? Okay, next one. What is the meaning of ER? Endoplasmic reticulum. Very good. Okay, endoplasmic reticulum mainly divided into two. Smooth endoplasmic reticulum and rough endoplasmic reticulum. The main difference between smooth and rough endoplasmic reticulum. Rough endoplasmic reticulum contains ribosomes in their membrane. Here, No ribosome. Other than that, they are the structural different. Rough endoplasmic reticulums are flattered cell. Flattered sex. Flattered sex. Okay? Sex. What is meant by sex? Bag. Bag-like structure. Okay? Smooth endoplasmic reticulum. Tubular sex. Tubular sex. This is the main difference between smooth and rough endoplasmic reticulum. Smooth endoplasmic reticulum made up of tubular sex. Rough endoplasmic reticulum is made up of flattered sex. Flattered sex contains ribosome in their membrane. Okay? Which type of ribosome? 70S or 80S? 80S, why? Because these endoplasmic reticulums are present only in eukaryotes. So eukaryotes contain 80S ribosome in their membrane. What is meant by vesicles? Normally, what is the main area of rRNA? What's the main area of rRNA? Main production area. rRNA. Nucleolus. Okay? Nucleolus producing rRNA. So, how it travels? They need one packaging-like structure. Okay? Before going to the cytoplasm, because cytoplasm is the fluid-like structure. So, before that, they need packaging-like structure. For that package only, we are telling vesicles. Okay? Vesicles. Next one. So how many functions? Transport protein synthesis, synthesize glycoprotein, produce transport vesicles, and synthesize membrane phospholipids. Only 4 functions. No.
Facilitates the growth of more membrane by adding proteins and carbohydrates. Therefore,
Okay, Golgi complex. Golgi complex. Golgi complex. Golgi complex also composed of flattered sex or tubular sex. Is it look like flattered or tubular? Flattered, okay. Flattered, okay. Tubular should be like this, no? But this one is flattered sex. There are 2 phases. This one, cis phase. Opposite one, trans phase. Between these 2 phases, there are pocket-like structures. Those structures are known as cristae. Okay? Cristae. Cis phase, trans phase, cristae. Flattered sex. Next one. The main function of Golgi complex, production of transport vesicles. Production of transport vesicles. That function is common for RER, SCR and Golgi complex. So, it produces transport vesicles. This phase is located near to the which part of the cell organelle? Didn't tell Willu? Didn't tell Willu? I think RER is here something. What? ER, yeah, just normal ER. Okay? This phase is located near to endoplasmic reticulum. So, cristae, transphase, cisphase. So, this is the simple structure of Golgi complex. Main function of Golgi complex, Production of transport vesicles and producing substances, packaging and distribution. Okay, first function. Collecting, packaging and distribution of materials, manufacturing cellulose and non-cellulose cell wall components. Okay, production of cellulose. Production of cellulose and non-cellulose. Example for non-cellulose cell wall components. Pectin, peptidoglycan, only two. Yeah. Next one. Produce lysosomes. Okay. Produce lysosomes. So under Golgi complex, around how many points are there? Around 20? No, not that much. Yeah. This pocket-like structure is known as crystane. Inside the crystane, what's the name of the empty space? Lumen. Okay. Lumen. So this is the pocket-like structure. Same lumen is present in RER and SCR, that empty space. Next one. So how many functions are there? Three functions. Production, packaging and distributing materials. Second one, production of lysosomes and production of cellulose and non-cellulose components. Example, pectin and peptidoglycan. Peptidoglycan. Clear? Okay, next one. Lysosome. Okay, what is the meaning of lysis? Breakdown. Okay. Rupture. What is meant by rupture? Cell rupture. Bone rupture. Bone rupture means that solid part turns into powder-like structure, completely destroying. So that's called lysis. So cell lysis means rupture of cell. RBC lysis, rupture of red blood cells. Okay, completely breakdown. So lysosomes, the main function of lysosome, rupture of cell. Which type of cell? Virus infected cells and old cell organelles. Is it good to keep the old cell organelles inside our cell? No. Why? What's the reason? Because lack of function. Okay, lack of function.